Today we are honored to host our guest speaker, Professor Yoshida. Professor Yoshida serves as the director of the Liver Tumor Translational Research Program at the University of Texas and holds the position of Associated Professor of Internal Medicine. He is a member of several professional organizations, including American Association for Cancer Research, the American Association for the Study of the Liver Disease, the American Gastrophysiological Association, and the American Society of Clinical Oncology. Through his career, Professor Yoshida has produced numerous publications, and of course, some of us already had the chance to read his works, of course, in liver cancer research. He has led various international important projects, such as the Precision Liver Cancer Prevention Consortium, and Professor Yoshida's expertise spans a broad spectrum, including molecular classification, cancer risk predictive molecular biomarkers, radiogenomics, molecular subtype-guided liver cancer therapies, liquid biopsy, just to mention some. Today, we have the chance to have here Professor Yoshida sharing his insights with us through his presentation titled Biomarkers for Risk Stratification and Chemoprevention of Hepatocellular Carcinoma. So, Professor Yoshida, if you're ready, the screen is yours. Thank you so much for being here today.
Yeah, you are very welcome, and thanks so much for your very kind introduction, and thanks again for having me. Let me share my presentation. Do you see my slide in the presentation mode?
Yes.
Yes.
Okay,
thank you.
Great. Thanks so much for confirming. Yeah, I will talk about biomarkers for risk stratification and chemoprevention of HCC. On behalf of my colleague at UT Southwestern in Dallas, Texas, USA. So here's my disclosure, which has no influence to my presentation. So let me start from this, global oncology cost over the past decade. As you see, it's more than doubled, almost tripled over the past just only 10 years. Mainly due to the new generation molecular targeted drugs and immunologic agents, which are very effective in some subset of patients, but very expensive. And this plot shows the change in cancer mortality in males and females over the past 9 decades, which shows if you focus on the last 3 decades on the right side, thanks to the new drugs, we are seeing the sharp decrease of mortality in both males and females. But still, cancer is a lethal disease. So you may think with these very expensive drugs, but still limited efficacy far from cure for all patients, you may say prevention is increasingly important. And we think HCC is a low-hanging fruit, a rational proof of concept to demonstrate the utility of prevention. So this shows the natural history of HCC development, which start from exposure to risk factors, including HBV, HCV. Nowadays, most of the patients are treated or on treatment with antivirals, or exposure to excess alcohol or dietary fat and sugar, or accompanying metabolic disorders. And the liver disease gradually progress, starting from chronic hepatitis, and then liver fibrosis gradually accumulate to cirrhosis, and a cancer develop, and highly frequently recurrent after surgical ablative therapies. And there is supposed to be subclinical stage, but we still don't know how it exactly looks like and how long it takes. And according to AACR white paper, preventive intervention can be classified into 3 levels. First, primary prevention. And now we have HCV vaccination, which is shown to be very effective in reducing HBV-related HCC incidence and mortality. In Taiwan, where HBV vaccination was studied nearly 4 decades ago, now we see almost a complete elimination of HBV-related HCC in children and young adults. And as for the unmet need, probably lifestyle modifications or something similar may be the possible intervention to prevent metabolic etiology-related HCC. Secondary prevention comes after exposure to the risk factors and the establishment of chronic liver diseases. And currently, we have regular screening or surveillance for early detection of HCC. And also, we know antiviral therapies substantially reduce HCC incidence for both HBV and HCV. And as for unmet need, chemoprevention, besides antiviral therapies, is still unmet need. And we are still not sure alcohol abstinence have what impact on alcohol-related HCC. And lifestyle modifications may have some role in preventing HCC with metabolic etiologies as a secondary prevention. And tertiary prevention aims to reduce the recurrence of cancer or improve mortality after initial diagnosis of cancer. And now we do regular recurrence monitoring. And the recent clinical trial shows a very promising result for neoadjuvant and adjuvant therapies. And we just saw the positive result from IMbrave 0450 atezolizumab for patients at high risk of ACC recurrence, just published end of last year. And our colleague, Adam Yop and David C. Chang was a part of the trial. So this opened up the new strategy for tertiary prevention of ACC. So in my today's talk, I will focus on the secondary prevention, overviewing what we are doing to meet the unmet need. I will talk about two things. First, ACC screening with individual risk-based approach. Two, ACC chemoprevention discovery with reverse engineering approach for secondary prevention. So so-called precision medicine approach for cancer screening is gaining more attention because of the availability of a variety of biomolecules, such as DNA variants, circulating nucleic acids, tumor cell, protein, metabolite, extracellular vesicles, so on and so forth, which can be measured in body fluid specimens, which is accessible non-invasively. And here I will talk about the challenge we are facing for ACC screening. So this shows the relative prevalence of ACC etiologies before 2010s, before we have a highly effective anti-ACV drugs. So the active ACV infection was a dominant etiology in developed world, and the active HBV infection was a dominant cause of Eastern and Southeastern Asia and Sub-Saharan Africa, and some alcohol and masudi. And after the new generation antivirals hit the market, now we see HCV is quickly shrinking, and the cured HCV is increasing. And most of the HBV infected patients are on antiviral and with a very good viral control. Alcohol steadily increasing, especially after the pandemic. And masudi is sharply rising and are now taking over the viral etiologies. So this impact how we screen ACC like this. So this is how the ACC screening used to look like when ACV cirrhosis was the dominant etiology. Annual ACC incidence is 3 to 5%. There is some variation. And these numbers of people are the number of people you need to screen to find out the 5 red people who develop cancer every year. This was cost effective. Therefore, justified as a screening intervention. And now, after the increase of cured ACV and MASUD, steatotic liver disease, the screening now looks like this. You need to screen these number of people to identify the same number of patients who develop ACC, which correspond to annual ACC incidence rate less than 1%. This is no longer cost effective. So you need to have a way to redefine the at-risk population to allocate the limited resources for ACC screening. So here the idea of risk stratified ACC screening kicks in. So you may consider applying the test to narrow down the population you want to focus on for the regular screening. And then you may classify the patients into the risk group in a different degree, which can be followed by the different strategy of screening, regular screening, utilizing the different modality. So this idea is, that getting more practical with the emergence of new technology, imaging-based methods, and also the biomarker like AMRI, abbreviated MRI, contrast-enhanced ultrasound, GALAD, and the cfDNA tests that are emerging, and also the EV-derived biomolecule likely hit the clinical market sometime soon. And also we did some mathematical simulation exercise. What is the impact utility of this idea? So in this scenario, you do ACC risk assessment upfront and classify the patients into, let's say, 3 groups: high, intermediate, and low risk group, and apply a different ACC screening modality. For example, you may think to apply MRI for high-risk patients and a state ultrasound for the intermediate and low-risk patients. And you can measure the measure cost effectiveness, which is called the incremental cost effectiveness ratio, or willingness to pay threshold. This is a additional amount of money you need to pay to gain one more quality-adjusted life year for a patient. And with this strategy, you need to pay $89,000 to extend the 1 year of survival. If you do MRI for high and intermediate risk people and do nothing for low risk people, this cost is significantly reduced. And if you use the simplified version of MRI just to tailor for the ACC screening, this cost is significantly reduced. And then you can keep trying the exercise like this, and that you can also try the emerging biomarkers such as GALAD and cfDNA. My clinical colleague Amit Singhal just started the prospective trial testing GALAD to ultrasound and AFP. And also, this simulation exercise told us that the risk biomarker should achieve hazard ratio greater than 2 to make this strategy cost effective. So this is the benchmark for our risk biomarker exploration. So what kind of biomarkers are out there? So you can see many in literature, so which represent the diverse classes of biomolecules. This is a very brief summary, and you can find the exhaustive list of biomarker candidates in literature in this hepatology review published last year. But briefly, there are many genes with the germline DNA variants, which is listed on the right top: gene expression signature in liver tissue, circulating DNA or RNA in plasma, serum, and even the urine, and circulating metabolites in serum and urine. And also more recently, microbiome-related biomarkers also proposed for possible association with ACC risk, which can be easily measured by 16S sequencing, which is a much easier to sequence human samples, and also the other method. So this is one example, the DNA variants, SNPs, single nucleotide polymorphism. So this is very easy to measure. So very promising candidate risk biomarker, not only for ACC, but any other diseases. And many SNPs has been tested solely or in combination. And in combination, which is often called as a polygenic risk score, PRS, or something like that. And this large study came out from UK Biobank assess what is the clinical utility of the SNP for assessment of ACC risk. So the y-axis shows a quantitative measure of prognostic performance, and each bar, which is accompanied with the acronym, represent clinical variable-based scores. Many of these should be familiar with you. And you see the darker part on the top of each bar, which indicate the additional information you can gain from assessing SNP over these clinical variable-based score. So the top performer on the left is APRI and FIB4 plus SNP. But if you see the contribution of the additional information of SNPs, you can see that this information is very, very limited or minimal. So this is another type of biomarker as an example where we are working on over the past decades or so, which is the more functional biomarker. So this is the liver tissue messenger RNA-based signature, which we call the prognostic liver signature, PLS. And the serum version of biomarker, prognostic liver secretome signature, PLSEC, which is interchangeable with each other, which classified the cirrhosis patients into high and low risk group like this, which predict the HCC incidence over 15 years of clinical follow-up. And I'd like to thank to Dr. San Giovanni, Dr. Yavalon, and Dr. Colombo of University of Milan, who significantly contributed to the development and validation of this biomarker. And if you look at the source of this risk signature, we think this is biologically very interesting and insightful. So we projected the genes, high-risk related genes and low-risk related genes in the signature onto the single cell profile of the liver. And interestingly, most of the high-risk genes, which is shown on the top half of this heat map, is coming from the variety of a non-parenchymal cell or endothelial cell, cholangiocyte, stellate cell, and also the liver resident and also the bone marrow derived immune cells as shown in the top, but not much from the hepatocyte. In contrast, low-risk genes are coming from the hepatocyte, but actually the half of the hepatocyte population, not all. So with these biomarker candidates, we want to assess which biomarker can be informative and have a clinical utility to improve the early detection of the HCC in the real patients. And this is the phases of cancer biomarker development, which is proposed by Pepe and colleagues at NCI under the framework of NCI Early Detection Research Network, EDRN, which is a nationwide NCI-supported effort to support the development of cancer diagnostic and the risk assessment biomarker. And they initially proposed this framework to guide the development and validation. So this includes the 5 phases. And phase 1 starts from preclinical exploration. So you name just a candidate biomarker without any specific estimation for the performance. Phase 2, clinical assay development. And here, in most of the case, in the setting of case control study, you optimize assay and algorithm and gain the preliminary performance estimate in the case control study setting. So vast majority of the biomarker candidates in literature are either in the phase 1 or 2. So phase 3, this is a huge bottleneck. This is called the prospective retrospective or PROV design, which is spelled out at the bottom, prospective specimen collection, retrospective blinded evaluation. So here you need to have a prospectively and carefully collected samples and for the biomarker validation purpose with a predefined protocol. So in this setting, you can test whatever promising biomarker candidate and gain the estimate of test performance, which is more close to the real target population for the cancer screening setting. Phase 4 prospective screening. And a phase 5 cancer control, assessment of cancer control at the population level. So in the US, there has been a huge effort taken, multi-center effort taken to develop the resources for the phase 3 HCC biomarker validation. And these 2 are the representative ones and almost nearly only ones. First is a statewide cohort, which is called THCCC, which stands for Texas HCC Consortium, now enrolled more than 2,500 cirrhosis patients from these institutions statewide. And the second one is the EDRN HEADS cohort, which stands for hepatocellular carcinoma early detection strategy study, which is assemble the patients from these 7 institutions in the U.S. Now, this consortium have more than 1,500 cirrhosis patients actively on the follow-up. And this is the first phase is just completed, and we just started the second phase of the EDRN HES cohort. Now it says the CBC cohort. So we had a chance to test a biomarker in these resources, and this is a snapshot of the result in a variety of the subgroup of the patients. So as I said in the earlier slide, so the hazard ratio 2 was our benchmark, and very encouragingly, we saw this benchmark was met in all subgroups we tested based on the patient sex or age or etiology and the liver disease severity and AFP at the bottom. So this interim result was presented at DDW 2022, and we will present the final result and the more further refinement of the score by integrating with the clinical risk score at DDW this year in May. And this is an idea we are playing with recently, which is etiology-adjusted HCC risk prediction. So yeah, I just showed the PLSEC met the benchmark of hazard ratio greater than 2 in all major HCC etiology, which is now validated in nearly 4,000 patients in the US and Asia. And the ongoing validation is the 2,000 additional patients currently ongoing in Europe and Asia with our industry partners. But we also see some variation across these etiologies. So we are currently playing with the idea of an etiology-specific adjustment with a plugin biomarker like this. And we published the plugin for muscle, which is published, and also the plugin for HCV cure in preparation, and a plugin for HBV and alcohol in development. So this is one example of MASUD plugin. So in this heat map and the Kaplan-Meier curve shows how the plugin marker performs. So in the etiology agnostic PSEC biomarker, we saw the very low HCC incidence of low risk group, but it was not 0, but it's around 0.5% or something. So there is some reluctance to say, the patient in low risk group don't need screening because a very, very small number of patients may develop HCC. But in this plugin, now we saw very encouragingly no HCC incidence in low risk group, as you see in the Kaplan-Meier curve. And by combining with the etiology agnostic PSEC and this MASUD plugin, we can further refine the risk prediction. So the low risk group remain low risk. Intermediate risk, which is shown in the gray line, shows some modest the incidence. And high risk group shows a strikingly higher the incidence. And now we are further validating this in a larger patient cohort. So with these biomarkers, we think we can now classify the patients according to the predicted HCC risk, which can be followed by the tailored screening approach. And now we have many tools like a GALA, cfDNA, abbreviated MRI, so on and so forth, to tailor the personalized, individualized HCC screening protocol. So let me move on to the second half, HCC chemoprevention discovery. So let me start from how challenging the chemoprevention is in general, not limited to liver. So this plot shows an example of chemoprevention clinical trial, testing selenium plus vitamin E for prostate cancer chemoprevention. So this trial enrolled more than 35,000 men, followed up longer than 7 years. and you saw no difference. This is a huge amount of effort with no positive result. You can find a similar example in the liver, which is a much smaller scale, but still substantial effort, non-trivial, testing a low-dose interferon. The first one is the trial in the US called HCC, and the bottom one is one in the Europe, EPIC-3. And both failed to show the clinical utility, justifiable clinical utility of low-dose interferon. And I can keep showing the unsuccessful example of chemoprevention strategy, such as a cycloretinoid, COX-2 inhibitor, mTOR inhibitor. So you can find the many numerous candidate targets and therapy in literature, and you can think of a plausible biology to prevent, and also the supporting experimental data coming from mouse or rat or cell culture model. But clinical translation, clinical validation is very challenging because of the again the huge demand for the clinical trial in terms of sample size and the follow-up time. Therefore, unsuccessful or almost impossible. So we thought, what if we flip the order, starting from a clinical cohort, which we called, like to call the reverse engineering approach, if you will. So that to do this, you need to have a archive sample or cohort with already completed follow-up. So we think this is a part is very critical, and you cannot start from just the hands-on samples of convenience, but well characterized and carefully for the patient cohort to have a very reliable cancer risk associated genes or proteins or pathways. So in this case, our biomarker PLS, PLSEC could be one candidate to play with this idea. And once you have a biomarkers, which is surely correlated with long-term cancer incidence, now you can move on to, with more confidence, to the experimental system to assess the mechanism of action, so on and so forth. And also another interesting observation we had in patients is this biomarker can be modulated by HCC preventive intervention. So now we know anti-HCV therapy substantially reduce HCC risk. On the other hand, some patients still develop HCC, even after the successful HCV clearance. And we had a chance to measure this biomarker over time at these 4 time points in patients who are treated with direct-acting antivirus for HCV. So the first time point is before antiviral, at the end of antiviral, and 4 weeks after completion of antiviral, and 48 weeks after completion of antiviral. And the y-axis measure the quantitative HCC risk level. So in patients free from HCC after achieving sustained virological response, SVR, over the 5 to 10 years of follow-up time after antiviral therapy, you see this biomarker-based HCC risk level decrease over time. In contrast, in patients who develop HCC within 5 years, even after successful HCV clearance, this biomarker-based HCC risk level is unchanged. So we thought this could be a promising surrogate endpoint to measure the chemopreventive effect of new candidate agents. And we could find a similar example in other types of intervention, bariatric surgery, for example. So several epidemiological studies suggesting that bariatric surgery is associated with lower HCC incidence by 70–80% in some studies. And we measured what is the magnitude of reduction of biomarker-based HCC risk by analyzing the molecular profile of the liver in patients who received bariatric surgery versus control. And we saw consistent corresponding reduction of biomarker-based risk level with the odds ratio 0.2, and also statins. So as you see in the many clinical papers, statins are suggested to be associated with lower HCC incidence. And some studies suggest that this effect is limited to lipophilic statin, but not in hydrophilic statin. And we did the same thing, measure the change in the ACC risk biomarker level. And inconsistent, we saw the reduction in lipophilic statin, but not for hydrophilic statin. But this is all based on the retrospective studies, so waiting for the prospective confirmation. So let me, in the rest of my talk, let me talk about the several example of possible chemopreventive intervention, which came out from the reverse engineering strategy. So 5 classes of agent, generic drugs, kinase inhibitor, dietary supplement, circulating bioactive lipid inhibitor, and the immune-targeted agent. So lipophilic statins. So my colleague, Dr. Thomas Baumert of the University of Strasbourg, developed a cell culture system in which the HCC risk biomarker is inducible by HCC etiology, such as ACV infection or fatty acid treatment, HBV infection, and alcohol treatment. And then you can test the effect of the drug on the ACC biomarker, risk biomarker. So here you are seeing, again, the y-axis is the ACC risk level measured by the biomarker. And the change effect by these different types of statins. And we found the one lipophilic statin, atorvastatin, shows that most striking effect to reduce the ACC risk level. And so this is a summary of the study that suggests that atorvastatin shows the more ACC reduction because of the targeting of additional pathways on top of YAP and AKT, which is shown on the right side in the red color, RX alpha, so on and so forth. So based on this, we just started the prospective phase 2 trial, which we call TORCH trial, with Dr. Ray Chang of MGH, with support from NCI, in which we enrolled child A cirrhosis patients, which are predicted as a high risk based on the PSA biomarker, and randomized into either atorvastatin versus or placebo, treated for 48 weeks. And we will measure the PSA biomarker before and after, and measure the change of the biomarker as a primary endpoint. And we will follow up the patients to confirm the HCC incidence is really reduced, and also that correlated with this surrogate endpoint. So we also are having another chance to test this idea for another class of statin. So this trial, called the rescue trial, is just started as well under the Liver Cirrhosis Network, which is funded by NIDDK, which is a multi-center consortium of the around 10 institutions nationwide in the US, which has two major goals. One is to create the cirrhosis prospective cohort, and the second is this rescue trial. This trial tests another statin, rosuvastatin. So this is a hydrophilic statin. And we just got a grant to measure this biomarker before and after the therapy. So so this study will be done together with Doctors Anna Maidil, Cindy Molam, Duke University, and Dr. Ray Chan, MGH. So here we will have an opportunity to compare lipophilic statin versus hydrophilic statin in the setting of prospective trial. Other generic drugs. So as I said in the earlier slide, we can induce this risk biomarker in cell culture model on dish, and they test the drugs. So in this setting, you can do the drug screening like this. So you see the name of the drugs on top, and the control on the most left shows the baseline status of the induction of the high-risk biomarker, which is shown as the orange color as a global status, and the high-risk gene induction is shown as the red color, and the suppression of the good prognosis or low-risk genes are suppressed as shown in the blue color. And followed by that, how these risk biomarkers changed by this drug treatment. And in the best case scenario, the high risk genes, which is turn from red to blue, and the low risk genes turn from blue to red. that means a high-risk gene is suppressed and a low-risk gene is induced. So this is a favorable modulation. And in this example of drug screening, what we found is two generic drugs, captopril and nizatidine, are among the top hits, which is standing out from these other agents. So nizatidine is a histamine receptor antagonist, which is used as an antacid. And we found, so this study was led by, again, my collaborator, Dr. Thomas Baumert, found that this pathway is touched, especially in the subset of a macrophage population in the liver. And by inhibiting this pathway, you can reduce the HCC incidence in the preclinical mouse model. And a second hit is captopril, which is an angiotensin converting enzyme ACE inhibitor, currently used as a drug for hypertension. And this is again led by Dr. Baumert, which found this pathway is a crosstalk with the EGFR kinase signaling pathway, and which promote ACC development. And use of captopril can interfere with this pathway crosstalk and reduce ACC in preclinical model. So both agents are generic drugs, are widely available, and we know that the toxicity adverse effect profile are very well tolerated. So we think either of them could be the promising candidate for next clinical trial. So kinase inhibitor. So again, this is another reverse engineering approach, starting from the biomarker. So in this biomarker, we found the EGF are one of the top pathways encoded in the high risk in this biomarker panel. And the long story short, we found this pathway's activation in hepatic myofibroblast, which is transdifferentiation from hepatic stellate cell, seems to be important and creating the cancer permissive tissue microenvironment. And this idea was tested in the rodent model of cirrhosis-driven HCC. So in this chemical-treated animal, fibrosis and gradually develop and reach to the stage of cirrhosis, then HCC develop. And right before HCC started to develop, these animals are treated with the EGFR inhibitor erlotinib. And as predicted, we saw a reduction of HCC nodule in a dose-dependent manner. So with this result, we just completed the phase 1 trial, short-term low-dose erlotinib. So in the cirrhosis patients who undergo surgical resection for any liver tumor, that we take a biopsy before surgery, and the erlotinib is given for 7 days, and these patients undergo surgical resection. And we had a paired liver tissue and also the blood. And the erlotinib was given at these 3 doses, 25, 50, and 75 milligram, which were far less compared to the FDA-approved chemotherapeutic dose, which is 150 milligram. So the lowest is the 1/6, and also the second lowest is the 1/3, and next is the half of the chemotherapeutic dose. And here is the result. The primary endpoint was a phospho-EGFR suppression, which was achieved at the lowest dose, 25 milligram. And also that the HCC risk signature, PLS, reversed at 50 milligram. And very encouragingly, we didn't see any severe adverse effect at 50 milligram. And the only mild skin rash was observed at 75 milligram, but not dose-limiting toxicity. So so this plot shows that you may say waterfall plot showing the kind of a dose-dependent effect on the risk biomarker. So based on this encouraging result, we are now planning the longer phase 2 trial with a placebo arm. So next, dietary supplement. So this is an agent we are interested in, which is EGCG. This is a green tea catechin, green tea extract, which reverse high-risk PLS, which, as you see in the right bottom, this is a green plot. And in this cirrhosis-driven HCC model, we saw reduction of the tumor nodule, and also the favorable change in the biochemistry in the liver, bilirubin reduced, ALP, AST, ALT reduced. So with this result, we are starting again the phase 2 trial with the funding from NCI, which we call CATCH-B, which stands for Cateching for Chemoprevention or HCC guided by Biomarker. So the study design is basically the same, but only one exception. So EGCG and green tea product is known to cause liver injury, and which was a concern, but a recent study from DILI Consortium, the Drug-Induced Liver Injury Consortium, identified one risk allele, which is predictive of green tea-associated liver injury, which is the HLA B3501. So we incorporate this finding in this inclusion-exclusion criteria. So that we will enroll the Charlie patients, high FIB4, high-risk PL-SEC, but without the presence of this risk HLA allele, and then randomize to the ECG and a placebo or a placebo arm, and we will measure the biomarker change again. Circulating bioactive lipid inhibitor. So this is another target we found by utilizing the reverse engineering approach. So in this case, we started from a meta-analysis of cirrhosis patients cohort, which include more than 500 patients, patients from Asia, Europe, and the US. The transcript are synthesized into one matrix, and these cirrhosis gene modules are teased out. So each dot represents one gene, and the line indicating the functional connection, and the color indicating the functional group, which we call module. And then now here we have a 31 gene module, which each of which are supposed to represent a different biology, such as the inflammation, carcinogenesis, and the other thing, like a fibrosis, so on and so forth. And what we found is one module at the center, which is shown the pink color, is the only cirrhosis gene module associated with the HCC incidence over 10, 15 years of clinical follow-up. And this pathway, LPA pathway, is one of the key pathway in this module, lysophosphatidic acid pathway. So what it is? So it's related to circulating bioactive lipids. So this cartoon shows a hepatocyte, which secrete the enzyme called autotaxin. And the LPC is supposed to, circulate in the blood in patient with steatotic liver disease or metabolic disorders, or patient who have excess dietary fat and sugar. And this circulating LPC is converted to LPA by autotaxin secreted from the hepatocyte. And this LPA bound to the receptor on the surface of hepatic myofibroblast, and believed to be, the contribute to creating the cancer-permissive tissue microenvironment. And we tested the pharmacological intervention to this pathway by using the receptor inhibitor in the rat model and the mouse model. And as predicted, we could reduce the tumor nodule and reverse the high-risk pattern of the signature to the low-risk pattern by the inhibitor. And also the, I'm not showing here, but also the autotaxin inhibitor as well. So we thought this could be the promising target. And also we saw the anti-fibrotic effect, so which also are relevant to liver disease. But earlier generation of the compound was just tested in lung fibrosis. But unfortunately, the liver toxicity was observed. So for that compound, the liver program is just terminated. But these compounds evolved into the next generation with believed to be low liver toxicity. So I'm still hopeful this pathway could be the target for HCC chemoprevention with a newer generation of compound. Okay, so the last example, immune-targeted agent. So I just mentioned atezolizumab could be, immune checkpoint blockade-based therapy could be a promising strategy for tertiary prevention, and probably also for the secondary prevention as well. But let me also talk about another approach targeting the innate immune cell, different cell types, dendritic cell, as a promising preclinical evidence. So in this example, we started from a meta-analysis of human single-cell transcriptome profiles data, as shown in the left plot. So in this case, each dot represents each one liver cell, which is composed with a variety of cell types, such as a hepatocyte, cholangiocyte, immune cells, so on and so forth, and which are numbered like this and also colored. And from this analysis, what we found is a EDO1 positive dendritic cell, which is a number 29, and also the PD1 positive CD8 T cell, supposed to be the exhausted T cell, has some interaction with each other, and contributing to the generation of the high-risk signature, our risk biomarker. And on the tissue, which looks like this. So on top, H&E staining showing the liver tissue from one patient with muscle, and a dotted line circling the inflamed portal tract. And also you see the massive lymphocyte infiltration. And that you can do the spatial molecular profiling on the tissue. And this circle indicating the molecular profile of this HCC risk signature. And the blue color is the low, and the red color indicating the high status of this risk biomarker. And you can see this high-risk gene signature induction restricted within this portal tract. And based on the prediction, these two immune cell type may be interacting each other to contribute to the generation of the high-risk status in the portal tract, which we call the molecular liver biopsy. We could confirm these two cell types are indeed co-localized in this portal tract. So the left is a CD11C positive, EDO1 positive dendritic cells, and the right is a CD8 positive and a PD1 positive cells. And you can also infer the actual interaction between these two cell types. So this yellow highlighted group is the representing the two immune cell subgroup. Single cell cluster 03 is a CD8 T cell. Single cell cluster AC29 is a EDO1 positive dendritic cells. And there are many ligand receptor interaction as inferred here. So this may be also the possible target to disrupt this, the cell type interaction, which may mitigate the HCC risk. So this idea was tested in a cell culture model again. This is a very convenient model developed by Dr. Barnard. So in this cell culture model, the high-risk signature is induced by the fatty acid treatment as a model of steatotic liver disease. And one small molecule compound, epacadostat, was tested, which is a small molecule inhibitor of EDO1 at these 3 doses. And the modulation of the gene signature was again similarly tested. So again, so this is a basal induction of the high-risk status by the free fatty acid. So the orange color indicating the induction of high-risk genes, and the green color indicating suppression of low-risk genes. And this high-risk pattern is reversed by this epacadostat treatment in a dose-dependent manner at these 3 doses, and it looks like saturated at the 25 micromolar, and then not much change at the 50 micromolar. And we recently confirmed this compound indeed reduced HCC incidence in the rat model, but interestingly, only in male rats, but not in female rats. And we are currently looking into the underlying biological mechanism to explain the effect, which was presented at the ASLD last November. So to summarize, HCC risk biomarkers enable individual risk-based HCC screening to improve early detection. Reverse engineering strategy utilizing clinical HCC risk biomarkers efficiently identifies clinically relevant diverse classes of chemoprevention agents. And we think HCC risk biomarkers are promising surrogate endpoints in RCTs for cost-effective evaluation of candidate chemoprevention agents. So lastly, I'd like to thank my lab and the collaborators, and also, yeah, I'd like to shout out the collaborators from University of Milan and also the funding sources. Thanks for your attention, and happy to take your question.
Thank you so much, Professor, for your presentation.